Okay, now that you are familiar with the process of photosynthesis, now we go back to look at how chloroplast enables the process of photosynthesis to occur, so structure to function. Okay, so a bit of background first. The cells that have more chloroplast are obviously cells that are found in the leaf, and they are found in the mesophyll tissues in the leaf, right? So there are two layers or two areas of mesophyll tissues, the palisade mesophyll and the spongy mesophyll. So the cells in these mesophyll tissues, the palisade mesophyll cells and the spongy mesophyll cells, are the cells that have more chloroplast. Another cell that has chloroplast are the gut cells of the stomata. So not all cells in the plants actually have chloroplast, or not as many chloroplasts. You imagine that root cells, for example, will have very little chloroplast, or very few chloroplasts, if any, right? Because they will never have light, right, underground. Okay, so now we do a bit of revision as to the structure of chloroplast, which we have learned last year when we learned organelles. Okay, so the liquid right inside the chloroplast is the stroma. Please don't say matrix. Don't mix up your stroma and matrix. Yeah, don't mix up chloroplast and mitochondria. Stroma. Then we have the double membrane of the chloroplast envelope. Right, so outer membrane, inner membrane. There's a very, very thin intermembrane space in between, but functionally it's not so significant, so we won't be looking at intermembrane space, unlike the mitochondria. Yeah. So also very seldom we'll individually highlight the outer or inner membrane. Usually we just call it the chloroplast envelope, right? Because functionally they are together. Right? Okay, so inside the stroma there are also the ribosomes, right? 70S ribosomes, the ones that is similar to the prokaryotes. We have the granum that is formed by the thylakoid membrane, right? So the thylakoid that forms the membrane found in the granum, and we also have the intergranal lamellae. That's also the thylakoid membrane. Actually, that's in between two grana. Right? So together they are referred to as the lamellae system. Right, so the lamellae system is like a separate third membrane system, right, in the chloroplast. Yeah, so you have the outer membrane, inner membrane, and then you have the thylakoid membranes or the lamellae system. Right, this is a starch grain. What is not shown would be some of the lipid droplets as well that can be found in the stroma. Right, so one by one, yeah. The chloroplast envelope, right, a double membrane, is important to regulate the movement of substances in and out of the chloroplast. Right? So ATP cannot leave the chloroplast because the chloroplast envelope is not permeable to ATP, for example. Right? Oxygen can leave, so oxygen is permeable. Rather, the envelope is permeable to oxygen. So that helps with compartmentalization and specialization, right? To maintain the optimal internal environment inside the stroma, right? As for the enzymes, especially those for Calvin cycle, to work. The thylakoid membranes or the lamellae system, right, are important because that's where the photosynthetic pigments are embedded, right? So the folding of the thylakoid membranes to form grana, right? The stacking of the membranes to form grana would increase the surface area, right, for attachment of more photosynthetic pigments. Yeah, the thylakoid membranes, right, also encloses the thylakoid space. So that's the compartmentalization of the thylakoid space, which will then allows for the storage of high concentration of H+, right, in the thylakoid space for the proton motive force to be generated. So I already mentioned some of these advantages, how to relate the structure to function. So again, the Lamellae system, the thylakoid membranes, are where the photosynthetic pigments are embedded. So that's the site of light-dependent reactions. The folding or the stacking of the membranes allows a large surface area for attachment of more photosynthetic pigments, as well as electron carriers for more ETCs and more ATP synthase as well. The electron carriers, because they are embedded onto the membrane, then they can be arranged in a particular sequence. If they are floating around, then there's no sequence that we can say, right? But they can be embedded in sequence. So that will form the specific sequence of electron carriers with decreasing energy level to form the ETC. And just now we mentioned the thylakoid space, right? So the compartmentalization of thylakoid space, because the thylakoid membrane is impermeable to H+, except through the ATP synthase, that will allow the setup of the high H+ concentration inside the thylakoid space to generate the proton motive force to drive chemiosmosis through the ATP synthase to make ATP. Yeah, so that's how the Lamellae system or thylakoid membranes are adapted for photosynthesis. Then the stroma, which is this matrix-like fluid or fluid-like matrix, but don't say matrix. Right? Stroma is stroma, matrix is matrix. So the stroma is this very dense liquid that has a lot of things dissolved in it. Some of them are the circular DNA, RNA like mRNA, tRNA, and 70S ribosomes. There are also enzymes for Calvin cycle. There are also chemicals, products of Calvin cycles, like our biomolecules, our acids, sugars, right? And then the sugars will go on to form starch, right? So there are starch granules or starch grains. And besides carbo, right, besides amino acids, lipids are also formed, right, from the triose phosphate or glucose phosphates. Right, so there are also lipid droplets in the stroma. Okay, so that's where the Calvin cycle occurs, because there are enzymes, right, for Calvin cycles. The stroma is a liquid, right? It surrounds the thylakoid membranes or Lamellae system. So the products from light-dependent reactions can easily diffuse into the stroma to drive Calvin cycle, and likewise, after Calvin cycle, when the ATP and reduced NADP are used, right, then the ADP, the inorganic phosphate, and the NADP can then be recycled back to the thylakoid membranes for light-dependent reactions to occur. Right, so the exchange of substances is easy because the stroma surrounds the thylakoid membranes. The stroma also offers the space for storage of the products from Calvin cycle. The sugars can be dissolved in it. The starch granules, the lipid droplets can be found in the stroma. The DNA, RNA, the 70S ribosomes function for synthesis of enzymes and proteins involved in photosynthesis. Okay, so this structure to function of chloroplast to photosynthesis.